So welcome everyone. Today we are hosting Hamutal Slovin, an associate professor of neurobiology at the Gonda Center for Brain Sciences at Bar Ilan University. Hamutal studied biology as an undergraduate here at Hebrew University, and he received her PhD at the physiology department at the medical school, also here at the Hebrew University, where she worked. Sorry for that. Mickey forgot to mute his microphone. So she worked for a PhD with Chagai Bergman and Ilan Vadia on Parkinson's disease. She then did her postdoc studies at the Weizmann Institute, but she pioneered the development of photosensitive dye imaging in awake behaving monkeys. And in 2004, she started her own lab at the Gonda Center at the Bar Ilan University. And her work focuses on how neuronal circuits in the primary visual area process, encode visual information, and also how eye movement involved in processing visual information in the brain. She combines electrophysiology with optical imaging-based photosensitive dyes, and her lab is among a handful of labs worldwide that use VSDI in awake behaving animals to study the brain. And the title of her talk today is Encoding Local Stimulus Attributes and Higher Visual Functions in V1 of Behaving Monkeys. Before we start, remind you that we encourage people to ask questions during the talk. Simply unmute yourself and ask your question. There will be also time for questions at the end of the talk. And without further ado, I welcome Hamutal to step in.
Okay, thank you, Didi, for the kind invitation. And please don't hesitate to interrupt me in the middle of the talk and ask your questions. And I hope that you can see the slide well as my mouse that is currently moving over the slide. Okay, so I'll start with a brief introduction on the tasks or the main tasks of the visual system. So as we all know, natural images are comprised from key features such as surfaces and contours. In fact, in order to process and interpret the information in images, the visual system needs to detect these contours and surfaces and combine them in order to create coherent objects. So for example, in this image over here, the visual system needs to detect the contour of this tube and then the contour of this color splash over here, the silver surface over here and the red surface over here, and then combine it together in order to generate the perception of a color tube. And the next thing that the visual system needs to do after generating the coherent object is actually to segregate it from the background. And in some cases, the segregation is really straightforward. So essentially over here, the visual system needs to segregate this color tube from the skies, from the view of the houses behind, the sea and the seashore, and so on. And in some cases, this figure ground segregation process is more difficult. For example, if we look over here, it's very difficult to notice that there is an awl over here. But once this animal actually turns its eyes into us, we can recognize there is an awl over here. Nevertheless, it is quite difficult to do here the figure ground segregation simply because the local stimulus attributes or the local features of this animal are very similar to that of the tree behind it. So in some cases, the figure ground segregation can be quite challenging. And the next step of the visual system, now that we have coherent object and we started this figure ground segregation and we segregated the object from the background, now the visual system needs also to segregate between individual objects. So for example, in this figure over here, we have here several object monkeys, which the visual system actually needs not only to segregate them from the hot bath they're taking, they're inside the bath, but also to segregate between the object themselves, namely between the monkeys. So to get to a situation where the features that belong to one object are not actually mixed with the features belonging to the second object. Oh, so my talk today, the outline of my talk today will be actually a according to these multiple tasks of the visual system. And I'll start with the encoding and decoding of surfaces and contours. I'd like to show you that encoding luminance surfaces actually hold unique features. And then I'll switch to encoding and reconstruction of shape contours at the sub-degree resolution when looking into the fovea of monkeys. The next topic in my talk will be electrical stimulation in the primary visual cortex. V1 is one of the main stages or area that were suggested to generate artificial vision. And this is also correlated with with our task in the lab to try to encode and reconstruct shape contour at a sub-degree resolution. And in the final part of my talk, I'll address perceptual processing, in this case, object and background and their relationship. And I'll deal with the two topics. The first one is figure from background segregation, and the other one is figure from figure segregation. So here is a very brief reminder about the visual pathway to the primary visual cortex. So light that is coming from an object that is located out there in the visual field is traveling through the lenses of the eye, falling on the back of our eyeball, on the retina, where it activates the ganglion cells that sends their information to the thalamus. From the thalamus, information is traveling through the optic radiation, arriving to the primary visual cortex, which will be the primary area I will speak about today. Now, the visual system in primate is actually comprised from several tens of different visual areas. And while there are many feed-forward connections between these areas, there's also an extensive feedback connection between this area, including into V1. And we believe that these feedback connections are deeply involved in higher visual processing, such as figure-ground segregation and figure-figure segregation, which I will refer to today. Okay. So in order to measure the brain activity, in our cases, we use monkeys and we use long-term voltage sensitive dye imaging. So very briefly, we have optical chambers, transparent optical chambers, through which we can image several visual areas. In this case, the primary visual area, secondary, and then area V4. And essentially what we do, we stain the cortex with voltage sensitive dyes. These are small organic molecules in our hands, which penetrates into the membrane of neurons and acts like molecular transducers, transducing changes in memory potential in these neurons into fluorescence. We can then pick this fluorescence using a sensitive camera, and essentially the voltage sensitive dye signal that we measure from each pixel in this image reflects the population response. Namely, this is the weighted sum of membrane potential from all neuronal elements from the upper layers, typically, of the visual cortex. It emphasizes subthreshold activity, but also reflects suprathreshold activity, namely spiking activity. The main advantage of this technique is that it offers high spatial, and by high spatial I refer to the mesoscale, namely tens of micrometer per pixel, and high temporal resolution, milliseconds per frame. And the specific parameters that we used are shown over here for those who are interested. Okay, so let me start with the first part, which is encoding luminance surfaces. I'd like to show you that we hold some unique features for this. So if we look into this nice picture of faces over here of this model, and we decrease the contrast, we can see that we gradually lose information. So we know that contrast or spatial contrast provides important information about the environment. V1 neurons are known to respond mainly to contrast, but less is known about the processing of positive, namely white, and negative, namely black, surfaces. And additional motivation for us to study the topic of surfaces is that we know that the perception of a surface is affected by its edges. So psychophysical studies have shown that the perceived brightness of a surface is influenced by its edges. Here's an example over here. So I hope that you can see these two circular patches, and the circular patch here on the left appear to be brighter than the circular patch here on the right. However, if I remove the background, you can clearly see that actually they have the same luminance level, the same also physical properties. So it is the effect at the edges of the surface generated by the background that is actually causing this brightness illusion. Another good example or interesting example is the corn sweet illusion, which I hope that you can see over here well. So we have here two surfaces, and the surface on the left appears to be brighter than the surface on the right. However, if I mask this central edge, you can see that these two surfaces are actually equal in their luminance level, so they look identical. And so the edge in the middle, and you can see over here the spatial profile of this unique edge, actually is generating this perceptual illusion. So the behavioral paradigm of the monkeys in this case is simple. They are simply fixating. Here is the fixation point over here. And then a white square appears or a black square appears over the screen. And we set to look what is happening in terms of the voltage sensitive dye signal. And here is the stimulus over here. And shortly after the onset of the visual stimulus, around 40 to 50 milliseconds after the onset of the visual stimulus, we can see here increased activation. Note that the brown and red colors over here are standing for depolarization or increased activity in terms of normal activity. And you can see that activity actually appears first at the edges of the square. There's a square-like shape over here, whereas in the middle, we can see a lower activity. And it looks like as time progresses, this hole in the middle seems to be filled in. If we present to the animal this white square over here, we can see similar response. Again, the response starts at the square projection over the primary visual cortex at the edges, and there's a clear hole in the middle, which again seems to fill in, but to a smaller extent than what we saw for the black stimulus. I brought here two movies that shows this effect. So this is the response to the black square. Over here, we will see time. This is the voltage sensitive dye response. Here we will see the stimulus coming or turning on. So let's see the movie. Here's the stimulus. You can see the activity starts at the edges and then information fills in. Let's see what is happening for the white square. So again, here it appears, and we can see the response started at the edges. There's a hole in the middle which filled to some extent, but not fully. And in order to quantify this, what we can do is we can define a region of interest at the edge of the surface, as you can see over here on the visual stimulus, which over the cortex will actually appear over here, and then at the center of the surface, which over the cortex will actually appear over here. And then we can plot the time course of response of the VSD signal, which appears over here. So we can see the response to the edge in green and to the center in red. And what we can clearly see is the response at the edge is rising faster and earlier than the response in the center, which seems to lag in or have a slower dynamics. So we wanted to better quantify this phenomenon, and what we did, we turned again into the average voltage sensitive dye map in the primary visual cortex. Here's a square which appears in V1, and then we took a spatial cut that is going through the two edges of the square as well as this middle part or the center part. And then, so what we did, we average across the width of this spatial profile. So this is the spatial profile over here, and this is time. And what we can clearly see is that shortly after the onset of the visual stimulus, we can see that activity increases at the edges of the square, and then it gradually fills in from the edges towards the center of the surface. And we can compute the spatial profile of this activation pattern. And from the slope over here, we can learn something about the mechanism of propagation of activity from the edges to the middle of the square. So what we see over here is that the achromatic squares, or these black and white squares, exhibit spatial propagation of activation from the surface edges to the center. And we can see that the average propagation speed is around 90 millimeters per second, which is well within the range of horizontal connection. Okay, so I'll move to the next topic, which is encoding and reconstruction of shape contour. Yes?
So the propagation speed is similar whether it's a black square or white square?
Yes. Yes.
Okay, thanks.
Sure. I'll continue to the next topic, which is encoding and reconstruction of shape contour sub-degree resolution. So what we wanted to do over here is to perform stimulus reconstruction, and this can be done actually at two different flavors. So we can do what is called reconstruction, which is to recreate the stimulus with the greatest details possible, namely pixel by pixel level, if you wish. And then there's another possibility to do this, and this is classification, which is actually find the image category or choose from a series candidate of images. And what we wanted to do in this study is to perform image reconstruction, namely pixel by pixel, via brain-inspired modeling without any image prior. So we are not choosing from any given set of prior images and from the voltage sensitive dye data. We have our paradigm is fixation. Here is the animal fixating on the fixation point over here. And this time we simply show the animal different shapes like circles and contours or different bars. And over here on the left, you can see examples for the stimuli that we showed the animal. The size of the stimuli are given over here. So these are within around 1 to 2 degrees. And these very fine contours or shape contours were presented at parafoveal location, as you can see from the eccentricities that are shown over here. And we wanted to know what is the voltage sensitive dye response. So for the circle over here, you can see that this is transformed to an elliptical shape in V1. These are average mapped across trials and at peak response. And for the small square over here, we can see a square-like shape in V1 with a hole in the middle. Whereas for this one, for example, over here, we see mainly a blob. Now, these are parafoveal location. We wanted to look into the fovea. So here is another animal. This time we actually imaged within the fovea. And so the shapes that we show over here are at the size of 0.8 degree, and the contours themselves are very thin. This is just 1 pixel, which is 1 over 35 degree of degree, yeah. And these are the voltage sensitive dyes that we measure from V1. And again, what we can see is that the circle in the visual field is transformed into an elliptical shape. You can clearly see a hole in the middle. A triangle looks also like a triangle in V1. So this part is actually the head of the triangle, or this upper corner of the triangle. And the base over here is actually, or the activity over here, sorry, is actually the base of the triangle over here. And this is a rotated square. I think that you can see here a nice rotated square, and you can clearly notice the edges of the square. We then presented also letters, and here is the VSD maps for the letter A, which is shown over here, the letter N, and the letter L. We then wanted to use a very simple forward model that takes the visual stimulus, convolve it with a point spread function, uses a nonlinearity function, and then retinotopic transformation of the generated image into the cortical image. And then we wanted to predict the patterns that are generated by this model to the response that we actually measured in V1. Over here you can see the prediction, and in many cases the prediction looks very similar to the response that we measured in the visual cortex. Here are the predictions for the letters, and these are again the actual measured response. There's again a good correspondence between the prediction and the measured response in V1. I will not get into details of quantification. I'll just say briefly that in many cases the predictions are similar to the evoked patterns. for smaller shapes, as we go into a higher eccentricity, then we get a less accurate prediction. And there are some, I would say, special zone like the vertical meridian, where we see usually less activation, and so the observation there are always more problematic to estimate. And the correlation coefficient between the prediction and responses are given over here. This is for the average data across trial and time, and this is for the single trials. Obviously, at the single trials, because they have more noise level, the correlation is lower. The next thing that we wanted to do is actually use the inverse model. So now we would like to reconstruct the stimulus from the cortical data. So this is the VSD response in V1. Now we can apply an inverse retinotopic model. So we can switch back to the visual field, apply nonlinearity. So if you wish, this nonlinearity actually cuts out the subthreshold activity of the voltage sensitive dye and remains mainly with the spiking activity, deconvolve the point spread function, and get the reconstructed visual stimulus. And we can look into this specific example for over here. So this is the average VSD response in V1, and this is the reconstructed stimulus pixel by pixel, and this is the original stimulus. And clearly you can see here there's a good correlation between these two shapes. So the previous example was at parafovea location, and we wanted to look into the fovea. So we switched again into the fovea, applied this inverse model, and over here you can see the reconstructed stimulus. So this is a circle that was presented to the animal. This is pixel by pixel reconstruction, showing over here a circle, a triangle that was presented to the animal, and this is the reconstructed stimulus, looks like a triangle, and this is a rotated square. We can see once again over here a rotated square. We then ask whether we can see this or compute this reconstruction also at the single trial level, because that's why what I've showed you are reconstructions that are done on the averaged data, average across single trials. These are reconstruction from single trials, and what you can clearly see here is that in many cases the reconstruction is good, but in some cases we lose part of the visual stimulus. This of course has to do with the high variability that we see in the single trials. To quantify the reconstruction performance, we can perform pixel-wise correlation between the reconstructed stimulus and the original stimulus. This is for the average data. So you can see here the different stimuli size that we use. And these are the correlation coefficient. This is for the average data, and this is for the single trial. As expected for the single trials, the correlation are a little bit lower. We then tested the performance over time. So this is shown over here. And what we can see is that the correlation coefficient between the reconstructed stimulus and the original stimulus develops as function of time as expected, and arrives to a steady state something like 100 milliseconds after the visual stimulus onset.
Hamutal,
Yeah.
So can you say what is the size of your point spread function?
Yeah. So that's a very good question. So the point spread function, the size of it is usually several millimeters. And in my hands, or it is my feeling that the size of the point spread function is also a function of the eccentricity. The actual
of the time from the stimulus onset, right?
This is also true, yeah, but since we are looking always at around 100 or 150 milliseconds after the stimulus onset, so we can sort of, you know, decrease the range for the estimation of the point spread function. This is one thing, but I think a larger sort of input of variance is the eccentricity from the fovea. So the point spread function, at least in my feeling and the quantification that we did in the lab, is much larger at the fovea than at more peripheral locations.
So how does this fit with the size of a cortical column? So it's much bigger than a cortical column, right?
Absolutely, yeah. Much larger than a cortical column. It depends which cortical column, but yeah.
But Hamutal, there are fixational eye movements that might smear the image a little bit.
Yeah, so the interesting thing is that once the stimulus appears, there's this phenomenon which is called microsaccade inhibition. And so once you get into this microsaccade inhibition, it lasts something like 200 milliseconds. So once the stimulus is turned on, at least the fast sort of like fixational eye movement do not appear for 200 milliseconds. So the effects of microsaccade in this case are completely minimal. This is one thing. The other thing, we also verified that we didn't have, by chance microsaccade in this time window. And the interesting question, Rafi, in relation to your point is actually what happens with the drift. And this is actually something that we are trying to understand now. The effect of drift in the visual cortex. Does it affect in a similar manner to the microsaccade? But at least I can say for sure that during these 200 milliseconds, where we usually analyze the stimulus, the effect of microsaccade is negligible.
Okay, thanks.
Okay, so then we wanted to add a temporal dimension. So usually a stimulus appears in the screen for a certain amount of time, as we show over here, the triangle, and then the neural response, the VSDI response, appears also as function of time. This is also true, for example, for a circle that you see over here. And now we'll see a circle contour that appears in the visual cortex. So what we did, we added the temporal kernel, and now we can look into the response and prediction over the cortex as function of time, as shown over here. This is for the triangle example. These are the detailed voltage sensitive dye maps. These are the measured, these are the predicted when we add the temporal kernel. And these are the spatial correlation between specific voltage sensitive dice frames and the specific predicted frames. So you can see here the correlation is function of time. And the temporal correlation is done between individual pixels. And over here you can see the values of correlation for this specific session and the grand average for across multiple sessions. We also wanted to do this temporal kernel for the inverse direction. So looking into the stimulus and reconstruction. So this is shown over here that we can code out also the time duration. This is the actual map. So these are the specific frames or time duration of frames, triangle stimulus that we actually showed on the screen. And these are the decoded or if you wish, reconstructed simulized function of time. And the correlation coefficient, again, appears over here. Now, usually in the real world, most of the stimuli are not really stationary. So we went to the next step, and this is adding a motion-like paradigm, if you wish. So what we did, we presented a sequence of stimuli going from parafoveal allocation towards foveal allocation. So we added a sequence of stimuli. This is shown over here, a circle, and then a rotated square. And what we expect to see over the cortex, I'll show it shortly, we expect to see how activity propagates from parafoveal allocation towards foveal allocation. So this is essentially what we see over the cortex, a circle, and then again, a rotated square. And what we wanted to do next, is actually compare the response to the prediction of the stationary encoder. So this is the spatial encoder of the shape itself together with the temporal encoder. And over here, what we can see is the response and the prediction in the primary visual cortex. And finally, we are heading towards more realistic stimuli where we have real motion and measuring also the neural response. And hopefully, we will show in the future the predicted response. So here's an example for a moving stimuli and a moving object in the visual cortex. Okay, the last point that I want to refer to in this project has to do with rodents. So recently, rodents became very popular in visual neuroscience. These are very important animal models because they offer many advantages. For example, in the mouse, one can easily do optogenetics experiment much easier than in monkeys. And importantly, you can see several visual areas simultaneously in the mouse cortex. So these are very convenient animals to work with. And so what we wanted to do is actually look into the mouse visual cortex and then present this animal with very large contours. And we were wondering whether we will see something that is similar to what we see in the fovea of monkeys. So this is just a reminder of the maps that we got in monkeys at the fovea. And over here, we have a slightly anesthetized mouse. And we first did retinotopic mapping. So what we did, we presented small squares in different location in V1, and you can see here the activation in V1. So in mice, this is the visual cortex, and these are the extrastriate areas, the lateral one and the medial ones. So we will focus mainly on V1. And then we presented to this animal shape at the size of 40 degrees, which are shown over here. These shapes over here are 0.8 degrees. And over here, you can see the development is function of time of the voltage sensitive dye response. But I think that already from here, you can get the impression that the patterns are very similar. Now we can put them, we can put the average VSD maps in the mouse and compare them to what we see in the monkeys. And while in the monkeys, we can see completely different patterns in the mouse, we can see similar patterns across the different shapes. And interestingly, if you look here in the middle, by the way, these 4 points over here are marking the borders of this large square that I mentioned. This is the large square of 40 degrees, yeah? Interestingly, what we can see here in the middle is that we have a large activation. Actually, the largest activation of this pattern is in the middle, where one would actually expect a hole. And we can quantify this over here using a ring analysis, and it shows that in the middle of this pattern, we get the highest activation. And so I think that I will conclude this part. I'll be happy to discuss the implication of this later on when we have the time for questions. But let me now move into the intermediate summary of this part. So for surfaces encoding, what we show is that at early times, these are edge dominated, and there's a hole in the center. There's evidence for neural filling in that propagate from the edges to the center, and the propagation speed is well within the range of horizontal connection, which suggests that maybe information is indeed propagating from the edges towards the surface, and this is how the edges of the surface affect the perception of the surface itself. Regarding encoding shape contour and reconstruction, what we show is reconstruction of shape contours via brain-like modeling. I showed you high resolution at the sub-degree reconstruction of imaging of images via cortical data. The current model is clearly very simplified, but it also encodes well very simple shapes. Over here, for example, we have the word Gandalf, which you can see the activation in the primary visual cortex. Temporal kernel, as I showed you, is in progress, and I think these have some implication for generation of the virtual vision, which again, I'll be happy to discuss later on during questions. I'd like them to switch into electrical stimulation in V1. And we know that electrical stimulation in the occipital lobe, particularly in the primary visual cortex of humans and monkeys, causes the appearance of phosphine, which is the visual perception of a small point of light. And electrical stimulation has been done already several tens years ago. He's a famous patient by Dubel, which had chronic electrode in the occipital lobe. And actually, in the past year, two very influential papers appeared where they showed that electrical stimulation can actually lead into a contour recognition, both in humans and in monkeys. And our motivation was that while there's an extensive body of research regarding the behavioral effects of electrical stimulation, little is known about the effect of ICMS on spatiotemporal patterns in V1 as well as in other area. And so the research question that we wanted to ask is, what are the spatio-temporal patterns of neuronal responses evoked by ICMS? And what is the relation to the responses evoked by visual stimulus? So if we want to use ICMS for the generation of artificial vision, we probably want to see how much is it similar to visual stimulus, and also what is the relation to saccade generation. So this time we combined voltage sensitive dyes together with microstimulation. Over here you can see a microelectrode that is inserted into the primary visual cortex. The protocol of ICMS is shown over here. And for a small visual stimulus that sort of like can mimic a phosphine if you wish, we use the small Gabor. Again, the parameters are shown over here. And so let me start with the results. The electrode was inserted here in the upper layers of this animal, and we started microstimulation, and you can see that activity is spreading quite fast around this electrode, covering an area of several millimeters. This is the average voltage sensitive dye map around peak response. And if we look at the time course of response, we can see that immediately following ICMS, we have increased activity. This is unlike the response to visual stimulus, of course, because you have the delay of information propagation from the retina to V1. But essentially what we see is that during the entire stimulation, activity grows and stabilizes at some point, and then following microstimulation offset, it decreases slowly to baseline. In this animal, the microelectrode was inserted over here in V1. However, in this animal, we were able to see several visual areas simultaneously, so V1, V2, and then also V4. And what we can see here is that following ICMS onset, we can see a clear patch of activation in V1, but also in V2. And then later on, again, activity is propagating within V1 and also in V2. And if we look at the average voltage sensitive dye map, which is shown over here, and we can mark these different patches in V1, V2, and V4, and look into the time course, we can see a similar thing that we saw in the previous example. Activity grows fast in the first step, then it grows steadily, and then when the stimulation turned off, the activity drops slowly to baseline. Interestingly, what we can see in this example is that ICMS evoked activity is propagating along the hierarchical pathway. This was an issue that was actually debated for several years. It was suggested, for example, by the Logothetis group that actually the propagation is sort of like not continuing, following one area after the one synapse connection after the stimulated region. Here we just wanted to show that the stimulation length or train duration length is actually related to the time to peak. So there's a linear relationship between the time to peak response as shown over here versus the stimulation. Now we would like to compare between the visual stimulus, between responses to the visual stimulus and the ICMS. So here is a response to a visual stimulus. This is the small Gabor. And what we can clearly see here is that after the onset of the visual stimulus with a certain delay, we can see a patch of activation very similar to what we saw in the ICMS, which is spreading over the primary visual cortex. And once again, when the stimulus is turned off, we can see that activity is going back into the into baseline. Over here, you can see V1 and V2. The only reason that you don't see here a patch in V2 is simply because the stimulus appear a little bit further away from the vertical meridian. So essentially, the patch in V2 is buried in the lumen sulcus. This is another example where it shows that a small stimulus actually generates patches both in V1, and this is V2. We then wanted to compare the time course dynamic in the ICMS and visual stimulation. So what we did, we looked into the rising phase of the ICMS and the visual stimulus, both the rising phase and the falling phase, and we aligned the rising phase on the response onset. And what we can clearly see is that for the ICMS, the rising phase is faster, if you wish. The dynamics is faster relative to visual stimulation. I don't think this is surprising. This is simply due to the fact that when you do microstimulation, you synchronize a large population around the microelectrode at the sub-millisecond resolution. And for the following phase, we see something that is similar. So once we stop the ICMS, activity drops down much faster than what we see for the visual stimulus. So it looks like the ICMS is inducing faster response dynamics. Now we wanted to see what is actually happening in terms of spatial propagation. So what we computed here are the derivative voltage sensitive dye maps. So these are the derivative maps. And then we took spatial profile through these patches of activation. And this is the visual stimulus condition. This is the ICMS. And for the visual stimulus condition, this is again space or the spatial profile. And this is the amplitude of the derivative. And in different colors, we can see the progress across time. What we can clearly see is that first we have this hill, which is growing up. This is as the stimulus, as the response goes up, and then activity drops down in a very homogeneous manner. However, if we look into the ICMS, we can see a different picture. So essentially what we see is that initially the response indeed is increasing as a hill, but then it drops down in a way that shows a lower activity at the microelectrode location, which over here, whereas at the edges of this activation, activity is still propagating. And we can quantify this by looking or by measuring the activation at the center, and actually the derivative value at the center versus the derivative value at the edges. This is shown over here. And you can see here two different clusters, one for the ICMS, which shows that the derivative at the edges is always higher than at the center, whereas for the visual stimulus, it appears the opposite way. So it looks like the ICMS is generating more like a wave-like propagation of activity within V1 as compared to the visual stimulus. The last thing that we wanted to do in this relation, in this topic, is to study the relation between V1 response and saccade generation. So it's well known that microstimulation in V1 in monkeys evokes saccades directed to the receptive field location of the stimulated cortical area. So this is taken from one of the Hovnik papers, where he stimulated in the primary visual cortex. So this is the receptive field of the stimulated neurons, and these are the saccades that are generated by the animal. And we looked into our session, and although our animals were actually trained to maintain fixation, we saw that in some session, indeed, the animal was making a saccade towards the receptive fields of the stimulated neuron. This is shown in this figure over here. So this is the horizontal position of the eye, and this is the vertical position of the eye. And in the unstimulated trial, you can see that the animal is holding fixation quite tightly. Here's another example from a different animal. This time we can see that the animal is actually making a saccade to the other lower quadrant of the visual field. And this is simply has to do with the fact that we microstimulated in the other hemisphere. When we look into the distribution of saccade size, we can see that the distribution is narrow, namely that most of the saccade had sort of like similar landing position, but in terms of latency, we can see that the latency is quite variable. And the next thing that we wanted to do is actually understand what is happening in trials where the animal is actually making a saccade towards the activated region within the retinotopic map or the receptive fields, if you wish, versus the trials where the animal did not make a saccade. Nevertheless, note that in both cases, the animal got the ICMS stimulation. And this is shown over here. So over here, we can see the VSD response for trials with saccade versus no saccades. And what you can clearly see here is that the response to the response in trials with saccade shows a higher activation. So you see here more white patches and a larger activation that is extending over more pixels or more millimeters in the primary visual cortex. This is the time of response in V1. In trials where the animal make a saccadic eye movement versus it was stimulated without saccades. And again, we can see that there is an activity difference between these two types of trials. This is the grand average in 2 monkeys, so showing essentially a similar phenomena. We also wanted to check whether this difference between trials with saccade to the receptive fields versus trials without saccade to the receptive field, whether this difference actually emerges also in additional visual areas. So we looked into V1, V2, and V4. This is also shown in the maps over here. And what, again, you can sort of get the impression that for trials with saccades, you get higher activation both in V1, which is here, and in V2, which is here, and also in V4, which is here. Now, thinking about stimulation, so electrical stimulation is clearly effective in a highly important technique. However, if we want to use activation of specific neurons or specific population of neurons, we need to use optostimulation. Optostimulation can be applied in a much easier manner in rodent, and especially what we wanted to do, we wanted to combine optostimulation with voltage sensitive dyes. So we went to rodents again in order to combine these two techniques. And what what we see over here is an experiment in rat visual cortex where we injected a virus. And over here, we can see the expression of channel rhodopsin 2 over several millimeters in the primary visual cortex. And we applied optostimulation in this animal for a very short time, at about 10 milliseconds. And then we imaged the voltage sensitive dyes in V1 of this animal. You can see a large activation spreading throughout V1 of this animal. And over here, you can see the time course of response, the VSD response. We removed the first frames where we had a large optical artifact, and we can see that the response gradually goes to baseline following the optostimulation. Over here, you can see the extent of activation that we got over the cortex, which is corresponding to several millimeters. We then wanted to actually compare between optostimulation and ICMS. So we are again in rodent. This time we're in a different sensory cortex, the biocortex. And what we did over here, we used ICMS and also optostimulation in this animal. And in terms of the optostimulation, we actually did two different experiments. We did optostimulation while waiting only 4 weeks after the viral injection, or waiting a longer time, which we call the long experiment. And first we found out that if you wait longer, you get more expression. This is not surprising. So this is, this case, this is the short experiment. This is the long experiment. So over here, you get much larger expression. But also if you optostimulate, what you can see is that you get much larger activation in terms of space if you wait a longer time after the viral injection. And in this sense, that was the case that was more similar to high frequency microcortical stimulation that is shown over here. We can quantify this by plotting the time course of response. So in red, we can see the response to the ICMS. This is at the end of stimulation. And in blue over here, you can see the response to the short experiment where we waited only 3 to 4 weeks. And in blue over here, you can see the response for the long experiment where we waited longer. So the ICMS is more similar to optostimulation if we wait longer time and let the expression grow larger. Okay, this is additional quantification in space. I will not get into this, but essentially it shows high similarity between ICMS and optostimulation for the case where we let the expression continue over several weeks. Okay, so let me summarize quickly this part. The neural responses evoked by ICMS show the spatial spread of few millimeters, which is much larger than the expected by the current spread and direct neural activation. And ICMS evoked responses that propagated along the hierarchical pathway to the extrastriate cortex, V2 and V4. And ICMS and visually evoked responses showed similar properties with some differences, with the ICMS showing faster dynamics and more like a wave-like propagation of activity. If we compare the size of activation over the primary visual cortex for ICMS And for visual stimulation, we currently have the estimation that ICMS, at least in our cases, generated phosphine at the size of 0.5 degree. And finally, I've showed you that ICMS evoked saccades that were directed to the receptive field of the stimulation site, and the population response in these trials were higher than in the trials without saccades. And I also showed you some similarities between optostimulation and ICMS, which depends also on the channel expression across cortical surface. So I'll switch very briefly to the last part of my talk, which has to do with perceptual processing related to objects and background. So I'll start with the figure from background segregation. And we wanted to address this question by looking into contour integration, which is a process in which similarly oriented elements are combined together to, or grouped together in order to form a coherent contour, for example, as shown over here. So this is an example where we have multiple gabors that, although they are not physically connected, they band together to generate a circular contour. And if you embed this circular contour in a noisy background of randomly oriented and positioned gabors, nevertheless, you still get this figure ground segregation. So this is, for example, a more difficult figure ground segregation that is actually challenging the primary visual cortex. So now we wanted to ask, what are the neuronal mechanisms underlying the figure from background segregation? Animals were trained on a detection task, so they needed to detect a contour in images that included a contour versus non-contour images. And essentially over here, you can see the vortex sensitive dye response, and there's two take-home messages over here. First, if we look at the early time, at early maps, this is 60 to 80 milliseconds after visual stimulus, you can see here clear patches of activation, both in the contour and the non-contour condition. And these patches of activation appear in the circular area, which is corresponding to this part of the stimulus, which is part of the circular contour, and also in the background area, which is corresponding to these gabor patches, which is also imaged in this part of V1. So there's not much difference at early times. So at early times, we have simply encoding of the gabor element. However, a little bit later on, something like 60 milliseconds later on, the activation is completely changed. We get a completely different pattern. And what we see is we see enhanced activity in the circular area, in the object area, and suppressed activity in the background. This activity or this pattern is actually unique to the contour versus the non-contour condition. And now we can also define a figure ground measure, which is simply the response difference between the circle and the background when you compare the contour and non-contour. And we can compute this figure ground measure as function of time. This is shown over here. And what you can see is that this figure ground measure starts to develop 70 to 90 milliseconds after stimulus onset, but arrives to a maximum around 250 milliseconds. Please note that this figure ground segregation is actually comprised from both enhancement in the circle and suppression in the background. And these processing are much beyond the individual element. So this sort of like global behavior, if you wish. And the late time of appearance tells us that this is probably a top-down influence.
Hamutal?
Can. Yeah.
So this enhancement in some way depended on the angle between the gabors? I mean when they are collinear or? Yes,
absolutely. So in order to, yeah. So maybe I'll go back over here. So what defines actually this circular contour is the fact that the orientation of adjacent gabors is similar. Now, if you take this orientation of gabor and just rotate them randomly, so the orientation over here appears now randomly, then actually you use the perception of the entire circle over here. And this is exactly the image that you see over here. And this is also completely reflected in the enhancement. So the enhancement over here, you have the enhancement here in the angle.
If you change the angle together coherently, instead of having them collinear, having them at 90 degrees, you will still have a perception of a circle.
Yeah, so this is again a good question. So we will see the maximal enhanced activity mainly for the collinear condition. If you start to sort of play with the local orientation and the collinearity, you can still see sort of like the object, and you can still see enhancement, but this is to a lower degree.
So it means that the enhancement that you see is a combination of the local features and the global perception of an object, right?
Yeah, I think that the object is generated also by the local features. Yeah, it's completely affected by this as well. Let me show you the next study where actually, it's not the next study, but actually the next step, where actually what we do, we reduce the saliency of the contour. This is shown over here. Okay, so we start to gradually jitter the contour. This is shown over here. And here we can measure the psychophysical performance of the animal. So we ask the animal, do you recognize a contour or not? As we increase the orientation jitter, which means as we decrease the saliency of the contour. And as for humans, what we can see is that the probability of contour detection is actually decreasing for these two animals. And interestingly, in relation to your question, what we see here is that this figure ground measure also decreases with the together with the psychometric curve. So there's a high correlation between the psychometric curve and the neurometric curve.
Thanks.
Okay, so let me now get to the final few slides, and this is figure from figure segregation. So as I mentioned in my introduction, one of the tasks or one of the more complicated tasks of the visual system is also to perform not only figure from background segregation, but also figure segregation, which is the next step. So here is a figure, for example, and we already know that if we look into V1, we expect to see enhanced activity for this figure. However, what happens when we present two figures in a visual scene? Then we expect to have enhanced activity with similar amplitude level for these two bars. And then the question is, how do we differentiate between the two figures? Because I just told you that we can discriminate between a figure and a background. So if we have two figures, how can we discriminate them in V1? And so what we trained the animal over here is over a discrimination task where it needed to discriminate between an image which included two figures versus one figure. The two figures actually included two bars that are shown over here, and the one figure included what we call the connected stimulus. So these are the two bars with two connectors over here. And the stimulus itself was comprised from a random pattern of moving dots where the figures were generated by common motion in one direction, and the background actually had a common motion in the other direction. So essentially, the animal needed to discriminate between these two stimuli category. I will not get into the details, but we verified that this is not pattern recognition, but rather a more generalized behavioral paradigm. And importantly, when looking into the primary visual cortex, this is the part of the visual stimulus that we looked. So essentially what we saw are two bars. In one case, these two bars were corresponding to two figures, and in the other case, these two bars were actually corresponding to one figure. So the local features could not have affected this two figure versus one figure activity in V1. Very briefly, just to show you the main result, this is the retinotopic mapping of the upper bar, the lower bar, showing that the connectors don't have effect in V1. And the main results appears over here. So what we computed over here is the separate minus connected condition. So this is the case where the animal actually saw and reported that there are two bars versus the condition where the animal reported as just one figure over here. And what we can see over here is that we have higher activity in the top bar, lower activity at the bottom bar, and not much difference in the middle, which is the background between these two objects. This is further quantification of this, showing the distribution of pixels amplitude in this differential map, separate minus connected conditions, and showing that in the top ROI, top bar, we get higher amplitude, bottom, we get lower amplitude, and in the middle, we can see no much difference between these two conditions. And we then defined a figure measure, which is simply the response difference between the top figure and the bottom figure for this differential map. And we can plot it as function of time. And I think maybe as expected, it starts to develop after the figure ground segregation. So this is a little bit later after the figure ground segregation. So the system first needs to do figure ground segregation and then decides how many figures there are there. And this is the very final slide before the conclusion, just showing that we can take this separation task and actually control the saliency between two figures and one figure by controlling the direction of a moving dot over here, the connectors. So we can gradually shift between two figures to one figure. So over here, we have the motion direction difference between the connectors and bars. And when this difference is 0, this means that the animal was actually looking into one object. So the probability of detecting two bars is high over here, and it decreases as expected as we get the motion direction difference between the connectors and bar to grow into 0. This is in the two animals, and this is the neurometric curve for the figure measure, showing very good correlation to the psychometric curve. So figure measure is related to the separation saliency between two objects and one object, and also to the behavioral report of the animal. Okay, so I will conclude now the last part of my talk, and I'll say that population response in terms of figure ground segregation, population response of early time showed activation patches that are corresponding to the contour and background. Oops, sorry. To the contour and background individual element. The figure ground modulation started to develop 70 to 90 millisecond post-stimulus onset. And we saw increased activity in the figure, suppressed activity in the background, and we see different labeling in the figure and the background. And this late modulation was correlated with behavioral performance and contour saliency of the monkey's perceptual report. In terms of figure segregation, so we imaged V1 population responses that were evoked by the two bars that were comprising actually just one figure or two figures. So the local properties were similar among the two conditions. And what we saw is that in the separate condition, unlike in the connected one, the population response of one bar was enhanced while in the other it was simultaneously suppressed. This divergent pattern evolved around 200 millisecond post-stimulus onset, and it was correlated with stimulus separation saliency and behavioral report. So what we want to suggest is that V1 is involved in labeling of each figure for the subitization, for the subitizing condition. So subitizing means that both humans and monkeys can actually discriminate between 1 to up to 5 objects in a very fast and efficient manner. We think that this figure segregation actually can be mediated by also V1, and this is also related to perception. And we think that both figure ground and figure modulation suggest the involvement of top-down influences through feedback connection into V1. And I want to thank very much for the lab members who did this excellent work over several years. So Hadar Edelman, Ruiz, and Shani did the microstimulation study. Guy Tzurabel, Amit Babayov did the reconstruction and encoding model. Yves Menashe and Shani did the rodent study. Ruiz also did some of the rodent study, and Ariel Gilad did the last part in my talk, which are the behavioral part. And I'd like to thank you for your patience.
Thank you, Hamutal. We have time for a few quick questions. So maybe I can start with you, Hamutal. You said something when you showed us the result from rodents retinopathy. You said something, if you find a difference between rodents and primates, and maybe you can elaborate a little bit more about this difference.
You mean the data on the visual cortex?
Yeah.
Yeah. So for us, at least for me, it tells me that I think that mice solve visual scenes in a different manner, I think, than what we do in foveal region. So I think that what is happening with mice, since the activity patterns are very similar, I think that in order to discriminate between different images, or in our case, different contours, the animal needs to locate or find out local discrimination or local differences between the different visual stimuli, rather than, you know, grabbing the entire contour or the entire stimulus on the entire image that was presented to her, rather than getting the coherent object by itself, as we are doing, for example, or as monkeys are doing. So I think that all the very interesting recent studies that are showing that mice indeed can discriminate between different visual stimuli, I think this is probably can be reflected also, I guess, in cortical activity, but I think it is more pointing towards local discrimination rather than global object discrimination, at least in my eyes, or at least according to the analysis that we have done, which is something that I think that we should take into consideration. You know, understanding the visual system in mice and rodent is, again, it's a very important, I think, direction of research, and cortical mechanism clearly can be discovered in these animals. I think this is very important, but there are probably some differences in the way that visual processing is done in rodents and in monkeys and humans, and we should take this into consideration in the future.
Thanks. Any more questions?
Yeah, I have one. Hamutal, great talk, really impressive.
Thank you.
In the case of the stimulation that sometimes succeeded in eliciting saccades and sometimes failed, did you see any prediction, a signal that is arising before the successful one that is sort of might be predicting that this is going to be a successful? I'm talking about before the stimulation, not
It's a great question, because there were several studies now suggesting that ongoing activity and spontaneous activity, in sort of like traveling waves in the cortex or in the brain itself, can actually affect at different stages the perception and also decision level. So we haven't tested it, but I wouldn't be surprised at all if we will find that some activity before the ICMS or spontaneous activity or activity level just before the ICMS actually defined whether the animal made a saccadic eye movement to the target or not. Namely, it is possible that this, you know, small increased fluctuation in spontaneous activity just in that specific trial actually added this information, which caused threshold crossing, and so the animal, if you wish, saw the phosphine in a higher contrast, or it drew the attention of the animal more, and so the animal made the saccadic eye movement. So yeah, I think one should actually inspect this, and this is an interesting line of research.
But do you see any traveling waves during on a single trial level during those trials?
Of ICMS?
No, you see before the ICMS, do you see in the spontaneous activity before you deliver the max, do you see any traveling waves?
Interesting that you are asking. We have a different project that has to do with the eye movement, and we think that we see something that has to do with traveling waves and eye movement. Yeah, we're just at the beginning of this. Maybe if you invite me next year, I'll tell you about this.
Okay.
I have some questions. It's 3 questions, but they're rather short. So first of all, I wanted to ask about saccadic movement in your first set of experiments, the ones regarding resinotopic maps, of how dramatic was the, I don't know if you have any data on how dramatic was the effect of this unwanted saccadic movements. I assume that the monkey was trained to fixate on the fixation point in the center of the screen.
Yeah, so again, it's a good point, which actually was raised during the talk, and it's always relevant. So once a visual stimulus is turned off, there's a known phenomenon which is called microsaccadic inhibition. So usually what is happening is that when a visual stimulus appears, the animal actually stops from doing even microsaccades for 200 milliseconds. So the animal is fixating, and the main eye movement that you have during this period is actually a drift rather than microsaccade. So the effect of microsaccade on the processing that I've shown is probably zero, because we verified that there are no microsaccades. The effect of drift would be very interesting to test. I don't have the answer for this yet.
Okay, then in relation to this, probably, did you test if stimuli that are shown closer to the fovea have a better resolution when trying to identify those figures in the classifier?
Yeah, yeah. So indeed, if you approach closer to the fovea, you can clearly see that you have much more details in terms of the shape. Let me go into one of the figures that I've showed you before, just to maybe clarify this point further on. Yeah. So this is what you see at the fovea. So at the fovea, you can see at very high resolution, and the magnification factor also detects here very high spatial resolution. If you go to more parafovea location, I'll go just one slide before. So this is the same stimulus that I showed just the slide before. It is the same size, and you can see how it looks like. Okay, so yeah, the fovea has dramatic effect on the representation of this stimuli, and clearly on the classification ability. Yeah.
Okay, and finally, most of the prediction models regarding visual stimuli on humans have been done on like multivariate pattern analysis using fMRI. But I don't know if there has been something so like an experiment like you presented done in humans, maybe in epilepsy or brain tumor patients.
Yeah. So you mean if voltage sensitive dye experiments were done in humans?
Yes.
Yeah. So well, I don't think this kind of experiment actually can occur in human. Would be interesting to check if one can develop this direction in the future, but currently it cannot be done in humans. I just want to mention regarding fMRI, which is another very important technique, that two things in decoding and reconstruction studies in fMRI. First, usually they use prior images, which means that the reconstruction you are doing is not, in most cases, is not pixel by pixel reconstruction, but rather what we called image decoding, namely choosing the closest image to the brain activity that you measure, which is a completely different question than the one that we dealt with, okay? This is one thing. The other thing that we need to take into account is the time resolution, which in the fMRI is, of course, it is in the range of seconds. With the voltage sensitive dyes and electrophysiological recording and neurophysiological recording, this is within the millisecond resolution. Yeah. So this is another thing.
Thank you. Very nice results.
Thank you.
Okay, so we'll conclude here. Let's thank Hamutal for a fascinating talk, and thank you for participating.
Thank you very much.